Welcome, everyone, to the 2013 Penn State Lectures on the Frontiers of Science. These lectures are a free mini course for the general public that we are pleased to be able to provide to you through the generosity of the financial support of the Penn State Eberly College of Science, and also through the generosity of our speakers who volunteer their time to give these lectures to you. The theme of our lecture series this year is Your Genes, How They Contribute to Who You Are. Today's event is the third lecture in this year's series, and there will be 6 lectures in all in this year's series. Our speaker today is Dr. Eric Green. He is the director of the National Human Genome Research Institute at the National Institutes of Health. An award-winning scientist and a highly accomplished research director, Dr. Green was at the forefront from start to finish in the game-changing Human Genome Project, which now has grown to encompass the structure, function, and evolution of genomes. He also is an influential scientific leader. As a series editor of a widely used laboratory manual of genome analysis, as the co-editor of the Annual Review of Genomics and Human Genetics, and as the founding editor of a journal called Genome Research, and as the holder and director level positions at the National Institutes of Health since 1996. The title of his lecture today is Bringing Genomic Medicine into Focus. Please let's give a warm welcome to Dr. Eric Green.
Well, thank you very much for that introduction. It's a pleasure to be here, and I'm delighted to see so many of you come out on a Saturday morning, especially a brisk, cold Saturday morning, to learn about genomics and to give me a chance to tell you a little bit about these exciting developments that are taking place. Now, I would say just starting out in thinking about genomics and its relevance, I would tell you that, for a long time, the field of genomics, which I'm going to describe to you, was very much in the purview of biomedical researchers. This was a research endeavor, research exploration. I think increasingly in recent years that's expanded, and we are seeing genomics, as I'll describe to you, becoming relevant to healthcare professionals, actually almost all healthcare professionals. But the reason I'm so delighted to see all of you in the audience is that what I want to describe to you is the fact that genomics is increasingly becoming relevant to patients and friends and relatives of patients, which means it's becoming relevant to all of us. So what I want to do during the course of this lecture is to really cover 3 major things that'll describe to you this landscape of human genomics, which is both exciting and challenging at the same time. In doing so, I want to start by describing a little bit of the past. I think some historical perspective will be very important. I want to frame the major part of my talk around the present that I'll describe as a framework that was provided to you. If you didn't get when you came in, you can get when you're getting out a recent strategic plan for genomics that we published a couple of years ago that I think very much organizes our thinking about where we are in genomics. And then at the end of the talk, what I want to do is to take you into the future and to share with you some of my thoughts on where I think all of this is leading. Well, now recognizing a very heterogeneous audience, and I realize from what I've been told about these gatherings is that people come with different types of backgrounds. I realize not all of you are trained in genetics and genomics, and so we have to start somewhere. I want to provide a foundation for everyone. Let's just think about a few basics. The human body or all living system made up of cells. The human body has about 10 trillion cells. Of course, what orchestrates those cells is a blueprint, if you will. It's the human genetic blueprint that is carried in the nucleus of all 10 trillion or so of those cells. Interestingly, each of those different cell types use that blueprint in different ways. That's why we have brain cells and we have blood cells, but they're all have in their nucleus the same exact genetic blueprint called a genome. In the nucleus of those cells are these structures called chromosomes. And it is, of course, these chromosomes that are basically the suitcases that carry our genetic material from one cell to the next, from one generation to the next. And the information molecule that is responsible for carrying that biological information. That blueprint, if you will, of course, is this incredibly interesting molecule called DNA. Now, if I had to think about the history, I would say that probably the most important historical moment that really set up what I'm going to describe to you in genomics probably would have to go back to 1953, because it was Jim Watson and Francis Crick discovering the structure of DNA, this double helical structure, which has become the icon for DNA, that was really pivotal in gaining an understanding about how it was that DNA was the information molecule, the blueprint molecule, if you will, that encoded all the information necessary for life. And in fact, it was that discovery of DNA's double helix that really set into motion a whole series of scientific discoveries that in the decades that followed, that in many ways set up everything I'm going to describe to you today. For example, we learned in the 1960s, how was it that DNA encoded information necessary for making proteins? Proteins are the brick and mortars of our cells, and we now know from what we learned, the translation of what's called the genetic code, how it was that the letters within DNA actually encoded information for making those proteins, those building blocks, the brick and mortar. The 1970s and the 1980s saw advances in our ability to study DNA in the laboratory. In particular, the 1980s brought about the molecular biology revolution, where we learned how to clone DNA, how to isolate DNA in the laboratory and be able to manipulate it in ways to be able to study how it was that DNA actually functioned. And along the way, we also figured out the kinds of methods that would allow us to determine the actual composition and sequence of the building blocks within DNA. We learned how to sequence DNA. And of course, the reason we wanted to sequence DNA is we recognized that the DNA alphabet is actually incredibly simple. DNA basically consists of 4 different chemicals. We don't even say the full names of those chemicals, we just abbreviate them A, T, G, and C. And it is the order of those chemicals that encode the information necessary for life. It's the G, A, T, T, C, 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 A, 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 and so forth that has the code that dictates how a cell interprets it to make proteins to do various other things. The other thing we recognized by the 1980s was that the human genome itself is a finite problem. It basically consists of 3 billion of these G's, A's, T's, and C's. That's a finite problem. It's just 3 billion. So could we order the 3 billion letters across the human genome and have that as reference knowledge for then figuring out all other aspects of human biology? And we had developed the technical tools for isolating DNA and for sequencing DNA, and it became apparent that yes, it was possible that we could develop an approach that would allow us to determine the complete sequence of the 3 billion letters that make up the human genetic blueprint. And that led to the launching in October of 1990 of the Human Genome Project. This large, audacious effort that was basically biology's equivalent of the moonshot, putting a person on the moon. We would go in and we would sequence the human genome. This was an effort that was regarded at the time as a bit controversial. It would involve big science, big collaborative approaches, international in scope, many countries involved, and basically developing teams of scientists to go in and industrialize the process of sequencing DNA to be able to figure out what is the sequence of the human genome. I had just, as a personal level, I had just graduated medical school, actually an MD PhD program. I had just started my residency in pathology, and I had the opportunity to join the Human Genome Project on the front line as a trainee, and I did it. And I will tell you, it was one of those things where it was very clear you were involved in something incredibly unique and exhilarating. And I will also tell you that it was something that was absolutely terrifying because we really had no idea what we were doing. We just had this audacious goal, sequence the human genome. We had some cursory tools. We had to figure out how to do it. But it was so motivating to be a part of it that, in fact, lots of people got together, great minds put their ideas together, and the strategies came forth for mapping and sequencing the human genome. And remarkably, when you get people together, they do incredible things. And by June of 2000, just shy of 10 years after that, came an announcement. It was a big deal announcement involving the President of the United States, involving people over in the UK. Tony Blair was involved in the announcement at the time, that we had in fact developed a draft sequence of the human genome. It wasn't perfect, but it was pretty darn good, and it was regarded as a great celebratory moment. A few months later came a publication that basically described the first view of the draft sequence of the human genome, And in this issue of Nature is largely going to be regarded as one of the most important publications ever to come out of the biomedical research enterprise. It wasn't done, by the way, because I would certainly be the first to tell you that this was just a draft sequence, and in fact, there was still more work to be done, but we went back to the laboratories, finished up the sequence, made it really high quality. And then in April of 2003, precisely actually 50 years after that Watson-Crick discovery of the double helical structure of DNA, came the completion of the sequence of the human genome, and with it came an end to the Human Genome Project. That was about 9 3/4 years ago. Actually, it was 3,579 days ago that the Human Genome Project ended, for those of you keeping track of these things like me. It was truly remarkable that this had been accomplished, and it had been accomplished not in the estimated 15 years that was originally envisioned for the project, but rather we had finished the project in 13 years. Now, a lot has happened in genomics because the tools that have been developed for studying the genetic blueprint of humans could be used in various other ways. And I would say there's been many, many applications of genomics that have taken place in the last 9 3/4 years. I will just show you a subset of these that are certainly very relevant, where genomics has found its way into having a significant impact on these fields of study. That's not what I'm going to describe today, although these are all very interesting. In fact, there could be a talk in and of themselves in each of these areas. Coming from the National Institutes of Health and being trained as a physician, you might imagine that where my emphasis is going to be is on health, disease, and medicine. And in fact, I've been at the National Institutes of Health now for about 18 plus years, and we are funded by the federal government. We are part of the federal government. We have funding to really focus on using the discoveries that we make to improve human health. And I'm now the director of the National Human Genome Research Institute, and our institute was originally created to lead the US's effort in the Human Genome Project, but we now see our mission as advancing human health through genomics research. And that's the focus of what I wanted to describe to you today. And I would tell you in particular, as the director of this institute, and this institute is really the largest funding agency in the world dedicated to genomics research, I take very seriously what our mission needs to be. And having been wildly successful at the Human Genome Project, I think very much what we need to be focusing on is operationalizing this to actually improve human health. And the focus of what I'm going to describe to you is on genomic medicine, putting together the notion of genomics and clinical medicine, and really to encourage and facilitate what I regard genomic medicine to be, which is an emerging medical discipline that involves using an individual's genomic information as part of their clinical care. In other words, most of us trained as physicians, we treat patients fairly generically, one patient to the next, one human being to the next, but in fact, each of us has a unique genetic blueprint. And now with the tools of genomics, we have the potential to use that ability to have information about a patient's genome to eventually help tailor their medical care. So the way I look at this is a journey, if you will, to be able to accomplish something such as creating a medical discipline and really advancing human health through it. And that in many ways, that journey begins with the Human Genome Project. That was the starting line. It wasn't the end of anything. It was really the beginning of everything. And someday we want to see the realization of genomic medicine broadly defined. But this is a journey. It's going to involve many, many steps. I don't even pretend to know what all those steps are going to be at the moment. But we embark on a journey like this reasonably optimistic because we were so successful at the Human Genome Project. We've got to believe we're going to realize genomic medicine. And the reason we need to do this is it will fulfill the promise of why we sequenced the human genome in the first place. We convinced the funding agencies, we convinced the scientific community that the reason to have the Human Genome Project, it would provide foundational information about the human genetic blueprint that would allow us to eventually improve the way we treat patients. And it will improve human health as a result. And that is the promise that I think we need to now fulfill. So how do we sort of do this journey? What do we imagine this to be? Well, I will tell you that in thinking about this journey from the base pairs of the Genome Project to the bedside of patients, or if you prefer the metaphor, from the double helix to health, the day the Genome Project ended, literally the day the Genome Project ended in 2003, our institute published a strategic vision for what's next for genomics now that we have the first sequence of the human genome. And this was a remarkably valuable document that sort of cast out a series of ideas of what needed to be done. And that was 9 3/4 years ago, and I will tell you, as a co-author of that strategic plan, I thought, it would be forever before we actually accomplished some of the audacious things we described in that document. But what's happened in the last 9 3/4 years have been just truly remarkable. Once upon a time, we'd have graphic artists that would render illustrations like this that would marry the idea of clinical medicine and the double helix and the genome and think that someday, these images would be a reality. But I don't think 9 3/4 years ago, any of us thought it was going to be anything less than 20, 30 years. And yet, we developed new technologies, we generated remarkable amounts of data, and with that came incredible opportunities to actually make this a reality. And in fact, we found a few years ago ourselves poised to make this a reality in a far shorter period of time than we had ever envisioned 9 3/4 years ago. And so as a result of that, we recognized that it was time to put out a new vision because we had actually almost outlived our 2003 vision. And so a couple of years ago, we completed another strategic planning process that yielded a new strategic vision for genomics, one that we published then in February of 2011. Actually, we published it in the issue of Nature precisely 10 years after that publication of that first draft sequence of the human genome. Now, a reprint of this strategic plan was available when you came in. I hope some of you picked it up. If you did, maybe you could pick it up on your way out. Any of you who are interested in reading this and didn't get a reprint or want to send this to somebody, if you simply look at this URL, this is a URL where you can freely download a PDF version of this and share it. And I really encourage you to read this. This was not written for a specialist in the field. This was written to a very, very broad audience, and it really does describe in many, many ways the exciting opportunities of operationalizing genomics to make it a reality en route to a realization of genomic medicine. So I thought what I would do is to describe to you some of the key elements of the strategic plan, and then you'll see I will frame it because it'll give me an opportunity to review what's taken place over the last 9 3/4 years. And then we'll go into the future when I'm done with that. Interestingly, what we heard about several years ago before we wrote this new strategic plan in 2011 was that it was time to be more sophisticated and more specific in iterating the steps that were going to be needed to realize genomic medicine. And at the end of the day, we found it very useful to organize our thinking, and increasingly we organize our scientific programs, around 5 major domains of research activities that were going to be required. Let me introduce them to you. The first domain is to do research to understand the structure of genomes, how genomes are put together. Sounds a lot like the Human Genome Project, and in fact, this is what we're most familiar with. But you don't want to just know how the genome is put together, you want to know how it works. So you also then need to have research activities that help you understand the biology of genomes, how it is that genomes orchestrate that information that then confers biological function. Well, once you understand how genomes work, you might want to know how genomes can influence health and disease, and so you want to have research that helps you understand the biology of disease. And when we get to it, you'll hear about how virtually all disease has a genetic and genomic underpinning. Of course, if you know more about disease, it might give you opportunity to advance medical science. So you want to do research to be able to demonstrate that you can use genomics to advance the science of medicine or medical science. And of course, you can't just stop there, because just because you have a medical advance doesn't mean when you go to operationalize it in the healthcare delivery system, it'll actually do anything. So you also need to do research that actually demonstrates you improve the effectiveness of healthcare. So these 5 domains, in essence, become a framework by which we think about really everything we're doing en route to genomic medicine now. The other thing that these 5 domains are very helpful is they provide us an organizational framework for thinking about the last 20 years, and then later I'll be able to show you how we think about the next 20 years. Because we can sort of inventory where we've been successful and where all this is going. What do I mean by that? Well, for example, we could think about different eras of time and think about where there have been accomplishments and how they distribute across these 5 domains. So, for example, the Human Genome Project. Well, during the era of the Human Genome Project, if I simply took a blue dot and put it down, when a blue dot represents an accomplishment, a scientific accomplishment, just very generic, where would those blue dots pile up? And as they piled up on top of each other, they may even change colors because they'd be so dense. Basically, the bottom line is the Human Genome Project was almost exclusively about this first domain of activity. We used genomics to understand how genomes were put together. We sequenced the human genome and other genomes. Yeah, we learned a little bit about how genomes work, but fundamentally, the center of gravity was around the first domain. Well, what's happened in the last 9 3/4 years since the Genome Project ended? Well, we continue to explore genomes. We've continued to learn a lot about how genomes are put together. But increasingly, as I'll describe to you shortly, we've begun to understand how genomes work. Oh, sure, there have been some opportunities to look at diseases, but mostly those have been much less in terms of the number of accomplishments compared to these first two domains. So let me then drill down in detail and focus in for a while on the last 9 3/4 years. What are these accomplishments that have characterized the field of genomics since the end of the Human Genome Project? And what do these blue dots really represent? Well, what I want to tell you about are 5 major steps that we've made going from the base pairs of the Genome Project en route to genomic medicine. What are some of those steps are? Let's start with the first step. Well, the first step is to understand the function of the human genome sequence, understanding how the human genome works. Really, research activities within these first 2 domains, if you will. But why do we need to figure out the function of the human genome? Isn't that what the Human Genome Project did? No. What the Human Genome Project did was to produce this. Actually, this is only 0.0001% of what the Human Genome Project produced. This string of G's, A's, T's, and C's. I am fairly certain any of you staring at this cannot immediately interpret the biological information that it encodes. But it encodes biological information. We just don't know the grammar, we don't know the words, we don't know the syntax. But it's all there. We just got to figure it out. Well, I can also tell you that the day the Genome Project ended, our ability to actually interpret this incredibly complicated text was very, very infantile. We barely knew how to really interpret this at all. We had to work very hard. That's what we've done the last 9 3/4 years. What have we done? Well, the first thing we started at is that we did what we did well. We sequenced genomes, and we realized that there'd be a remarkable power in comparing our genome sequence, our genetic blueprint, to the genetic and genomic blueprints of other creatures. Let's remember, humans are just one little twig off this very complicated tree of life, but we're all interrelated. And the evolutionary process has been such that we all share a tremendous amount in common in our genetic blueprints. But the fact of the matter is we are unique, and wouldn't it be cool if we could get the genomic blueprints of these other creatures, compare them to ours, and then recognize things that are the same must be functionally important, because otherwise evolution would have changed those sequences, because that's what evolution does. And so off we went, immediately sequencing a series of other animals, including laboratory animals, companion animals, our closest relative. But we also realized it was very important to sample across this very complicated tree of life. And so we started sequencing other critters and recognized that there's a lot of statistical power associated with sampling across different parts of this tree of life, and then using computer programs to analyze and reanalyze all the different sequences to find those sequences that have never changed over large evolutionary distances, because those sequences for sure are functionally important, and it would start to teach us the words that we needed to interpret in the human genome sequence to understand how the genome actually functioned. What did we learn from some of those studies? By the way, we've now sequenced probably about 36 other vertebrates' genomes have now been sequenced, and we have lots of computer tools that help us interpret and reinterpret. We learned from that, for example, that across our 3 billion letters of the human genome, about 5% of them or so are highly conserved across virtually all mammals. In fact, they're incredibly highly conserved, and almost for certain those are functionally important. And that allows us to sort of highlight the parts of the genome that we want to study more intensively, and to be able to figure out those are the ones that are likely biologically important that we want to sort of look at more carefully. So for example, we could pull out one color highlighter, which turns out to highlight about 1.5% of the letters in our genome. And that highlighter corresponds to those sequences that directly code for protein coding genes. In other words, these are the sequences that actually encode the bricks and mortar in our cells. But it's only 1.5% of our genome. Now, it turns out that if 1.5% are these genes and 5% is highly conserved stuff, that leaves about 3.5% that we better highlight in something else. And we are highlighting that in something else. These are as conserved through evolution as genes in many cases, but they're doing things other than directly coding for proteins. Well, what are they doing? Well, we're learning. We do know that some of them are determining where and when genes get turned on and how much. I'll give you an example, and I sort of alluded to it earlier. Why is it that every one of our 10 trillion cells contain the gene for insulin? But insulin is only made in a subset of the cells of the pancreas. Well, that's because there's a switch that determines, hey, I'm an islet cell in the pancreas, I will turn on this one gene, and that switch is some of this purple stuff. So there's all this circuitry that determines where and when genes get turned on. Similarly, hemoglobin, every one of our cells contains the gene for hemoglobin, but hemoglobin only gets made in a subset of blood cells. Why is that? Because of the switches that determine when a gene gets turned on, and so forth. There's a lot of complexity there, and we're learning about that complexity. Oh, but by the way, it turns out that we now know from the last 9 3/4 years that it's not just the primary sequence of DNA that confers function, but there's other aspects of DNA that confers function, and that's something called epigenomics, because it turns out that our DNA gets decorated with chemicals and gets associated with other molecules, and that those little decorations that take place on our DNA also influence how our DNA works. That's called epigenomics, and it's some of the ways that environment influences us by influencing our DNA, and we now are learning a lot about the epigenomic landscape. And increasingly, we're developing better and better methods that allow us to read the second code, the epigenomic code, the decoration code on our DNA. And that's a whole additional code besides the primary sequence code. And a lot is happening in that arena as well. And in fact, we recognize the need to have a very concerted effort in interpreting the human genome sequence, the primary sequence, the epigenomic sequence. And our institute, for example, launched a project called the Encyclopedia for DNA Elements, or ENCODE, which basically was a large international effort to get investigators together who just focused on developing laboratory methods, computational methods, to critically interpret all of these information clues within the human genome, to get catalogs of all the genes, catalogs of all these other switches and circuits, catalogs of all the epigenomic changes in our DNA, and use that as a resource, if you will, to allow other scientists to understand how the human genome works. Last year was an exciting year for ENCODE. In one flurry of papers in these journals, they published about 30 papers. That was really the first comprehensive view of the functional catalog of elements in the human genome. And increasingly, such as shown on this little poster here, we're getting views. I'll show you a blow-up on this slide. Here are two regions of the human genome, and overwhelming amounts of information you can see from all these little marks and all these little lines that are called tracks. The details aren't important. I guess the analogy I would make is many of us have GPSs in our cars, and those GPSs might tell us where the roads are. That might tell you like the sequence. But increasingly, what ENCODE is giving us is information about what things are associated with different parts of our DNA, just like a GPS might say, oh, here's a road, but here's a gas station. Oh, and over here, here's a hotel, and here's a McDonald's, and so forth. So just like we are annotating additional information on GPS that gives us information about our surroundings, ENCODE is increasingly giving us information of what's relevant functionally across different stretches of human DNA. And this will continue into the future because it's actually even getting more complicated. We're also now learning that it's not just the linear sequence of letters in DNA that encodes information, and it's not just the decoration on DNA that encodes information, but we also now know that the DNA that sits in the nucleus of all of our cells is not so innocent. It actually has a three-dimensional conformation, and different stretches of DNA are interacting with other stretches of DNA as it's floating around. And in fact, that also confers biological information. And increasingly, we are learning how to read out that information and be able to interpret this three-dimensional information that also exists in our DNA. The fact of the matter is, we will be working on this for a long time. Decades from now, I predict, my children and then my grandchildren will still be interpreting and reinterpreting the human genome. Today, if you ask me where are we maybe at best have a Cliff Notes view of the human genome sequence, okay? You know, it's not like it's the last scholarly piece of this great novel. It is just the beginning superficial view, and for a long time, we will be trying to understand the complexities that reside in our 3 billion letters. So that's where we are on that first step. What about the second step? Well, the truth of the matter is we're not just interested in how a hypothetical human genome works. We want to know how each of our genomes work, because each of our genomes is just a teeny bit different. So we're interested in variation among our genomes. We're interested in knowing how we differ, and therefore how each of our genomes function in a slightly different way. These are research activities still mostly have been over the last 9 3/4 years in these first two domains of research activities. Now, what do I mean by that? Okay, well, what I mean by that, let's just sort of back up for a minute. Each of us has 2 genomes in us, right? We got 3 billion letters from mom. We got 3 billion letters from dad. 6 billion letters makes up a true individual's genome. Now, look to the person, look to the person to your left, the person to your right, and I will tell you that you differ from that person about 1 out of 1,000 places along the stretch of those 6 billion. Okay? And if you add it up, it's about 3 to 5 million differences between the person sitting next to you and you. And these are all variants, I'll indicate them here by these, that the great majority of which really have no biological consequence whatsoever. They're completely silent in the great majority of cases. But a subset of them are biologically important. Some of those, for example, might be problematic. They might be a ticking time bomb for some disease you might get or some other detrimental attribute. Some of those variants you have might be good variants. They might mean that you won't get a disease, or there'll be something else that'll be more positive. But by the way, each of you do not have a private 3 to 5 million variants that nobody else has. In fact, many, tens, actually hundreds of thousands of the variants that you have, somebody else in this auditorium has as well. And in fact, the problem of genetic variance is actually relatively finite, we believe. And in fact, one might imagine that one could catalog at least the more common variants that exist in different human populations. And that idea occurred to us. And so several initiatives were launched to try to understand human genetic variation, in other words, variants that exist within our genomes. One such effort, the Human HapMap Project, which had 3 major publications, started this process, and it aimed to do several things. First of all, it collected DNA from well-defined populations all across the globe, because we want to get this to be sort of a variant catalog of humanity, not just of any one population. The second thing it did was to use various fancy methods to figure out some of the most common variants that exist in all of those populations and make those available on the internet for all scientists to use. And then the third thing that this particular project did was to basically start to develop an architectural map, if you will, about how these variants relate to one another. And what do I mean physically along the chromosome? What I mean by that is it turns out that all of your variants, even when they're sitting really near each other on a chromosome, they don't sort of just go off in different directions every generation, but rather a given stretch of a human chromosome, a block of maybe 100,000 bases, might have 100 places in it that vary, but all 100 of those, that whole block tends to be inherited as a block, as a neighborhood from one generation to next. So they tend, and these are called haplotype blocks, and this is why this was called the HapMap Project, was to characterize these haplotype blocks for reasons that will become apparent in a few slides from now. There's a little scientific trick I want to teach you. Well, this was pretty good, but the fact is there was great interest in getting even more information about rare and rare variants, and so when better technologies became available, another international consortium was formed called the 1,000 Genomes Project, which initially aimed to catalog all the variants in 1,000 people, but of course, as always in genomics, we're overachievers, and now we're doing it over 2,500 genomes, but increasingly we are getting a richer and richer view of genomic variation that exists. And with these again, samples from across the globe, and through an initial pilot effort that was published in 2010, and then more recently, the first real critical look of the initial 1,000 individuals, we are getting remarkably deep catalogs of genomic variation. And with that is coming some insights about what individual human genomes look like. So let me digress for a minute, and let me tell you a few things about your genome. Your genome by the numbers. So I already told you your genome has about 6 billion nucleotides, 6 billion letters in it. I already told you that you have about 3 to 5 million places that a single nucleotide is different compared to, let's say, the person sitting next to you. There's also about 150,000 of your 3 to 5 million variants we have never seen before. Those are sort of new variants if we would sequence your genome. Most of them we've seen, but a few of them are relatively rare, and so we haven't seen them yet. And it turns out that about 60 of your variants neither of your parents had. Those are new oopses. Those are new typographical changes that took place in creating you. And so those are new. And then about another 10 to 20,000 places, not only do you just have a single nucleotide that's different, you might have a stretch of DNA that's missing, or a stretch that's inverted, or a stretch that there's a couple copies of something where in other people there's only one copy. Those are known as structural variants. Now you may also be asking, well, across my genome, how many places am I broken? How many places are things maybe not working the way they should, just in aggregate? There's probably about 100 places where a gene has been disruptive, should not be working. By the way, we have about 20,000 genes in our genome, and about 100 times you may have one of your 2 copies of your gene, because every gene you have 2 copies of. 100 times it might be disruptive, where one of your 2 copies is broken. Oh, and by the way, each one of us probably are carrying about 20 of our genes where both copies are broken. It's just sort of cool to think about. We have 20,000 genes, and each of us, on average, about 20 of those, neither copy works. That's why we're all a little different. And some of this has biological consequence, and some of it has less biological consequence. So that's a little bit of digression about your genome. But it makes you think about, well, which of these changes in my genome might have relevance to my health? Which ones predispose me to a disease? Which ones might influence which diseases I might get? Which brings me to the third step of accomplishment that I want to share with you. And here, we're starting to get closer to genomic medicine because we're starting to deal with human disease, and we're dealing with this middle research domain of understanding the biology of disease. There's great interest in understanding the genomic basis for human disease. So, to describe to you what's happened over the last 9 3/4 years, I need to drill a little bit more to explain to you about genetic diseases. All diseases are genetic. There's not a single disease practically that you can't name that I won't tell you at least there's a genetic influence on the severity of the disease, and many diseases have an overt genetic cause. But diseases can be largely divided into 2 broad categories, if you will, that their underlying architecture is a little bit different. Let me explain. And it's important to see the distinction because what's gone on in the last decade or so in research in these areas have been very different. On the one hand, there are rare genetic diseases. These are diseases like sickle cell disease, cystic fibrosis, Huntington's disease. Now, rare genetic diseases are rare, but they're also simple. And the reason why they're simple is because there's basically a defect in a single gene, one of the 20,000 genes in our genome, that is the major risk factor for getting the disease. These are otherwise known as monogenic, one gene. They're also known as Mendelian disorders, named after that famous geneticist Gregory Mendel. In his pea plants, he figured out about single genes, so they're called Mendelian disorders. Now, there can be other genetic variants that might influence the severity of disease, and there might even be some environmental influence on severity of disease, but by and large, rare genetic diseases are caused by defects in a single gene. Now, rare genetic diseases are devastating to patients and families, but they're rare, and they don't in aggregate represent the major healthcare burdens in the world. The major healthcare burden in the world are these diseases, because these are common diseases. These are diseases like hypertension, cardiovascular disease, diabetes, Alzheimer's disease, mental illness, many forms of cancer, and so forth. And common diseases, besides having a huge burden on human health, turn out to be genetically complex. And the reason they're genetically complex is every one of those diseases I just named are not caused by defects in a single gene, but rather it is a variance in multiple different places in the genome that together conspire with what is typically a larger influence of the environment to confer risk for getting the disease. It's not an absolute, it's an influence and it's a risk, and therefore it's more complicated because it is multigenic, otherwise known as non-Mendelian disorders. When the Genome Project was being envisioned, we always believed that having the sequence of the human genome would greatly accelerate the ability to identify the genetic basis of rare diseases. And there was always a raging debate about would we ever get good enough at figuring out the genetic basis of complex diseases. Let me tell you now, for each of these categories, what has happened over the last 9 3/4 years. So, shown here is a cumulative histogram of all of the single gene disorders or traits for which we have identified the genomic basis. So genomic basis known, and this is a cumulative histogram. Let me remind you, the Human Genome Project began there. The day the Human Genome Project began, there were only 68 diseases or traits for which we knew the genomic basis, 68. And you can't argue with the fact that the moment the Genome Project began, shortly thereafter, we saw a massive upswing in the number of diseases, single gene diseases, for which we figured out the defective gene. And the figure as of when I made the slide a couple weeks ago was 4,800. We went from knowing the genomic basis of 68 single gene disorders to now knowing the genetic basis of over 4,800. Clearly, that has been a remarkable advance in our understanding of the genetic basis of rare diseases. Now, that's pretty exciting, and it is a remarkable achievement, but you'll quickly learn from me, I'm a guy glass half full, glass half empty. The glass half full part of me says this is remarkable. We now know the genomic basis of 4,800 monogenic diseases and traits. The glass half empty part of me, though, is a reminder that there's still about 2,000 more single gene disorders that we don't yet know the gene. And on top of that, there's another 2,000 or so that we think a single gene is involved and we haven't figured out the gene either. So this is only about half done, and memorize this pie chart. I'm going to come back to it when I take you into the future. So that was single gene disorders. What about complex disorders, those multigenic disorders? Well, here I want to introduce you to a strategy and then tell you about remarkable advances that have taken place. There's something called Genome-Wide association study. That's a mouthful. And let me explain to you a few things. Shown here is a stretch of a human chromosome, and every one of these little black marks along the top represents a place that varies in the genome among people. These are known as single nucleotide polymorphisms. It's basically a letter that is different at that given position. Now, shown here in these inverted red triangles are these haplotype blocks I talked to you about. So again, this is a stretch of a chromosome, and let's say this block right there probably has maybe, I'm making it up, 100 of the variants that exist across that stretch. But all 100 of those variants tend to be inherited in block together as a neighborhood from one generation to the next. And the same for all the variants on this block, and the same for this one, and this one, and this one, and so forth. And so the idea was to do the following study. This is the study one wants to do. What you want to do is you want to take 1,000 people with a disease, let's say hypertension, and 1,000 people without that disease, without hypertension. And then you want to know the 1,000 people with hypertension, which variants do they have compared to those 1,000 people without hypertension. Now, you could type all 1,000 people for millions and millions of variants, but that would be incredibly expensive and impractical. But imagine if you just took a representative variant from each of these haplotype blocks, knowing that it was a surrogate for all other variants in that particular neighborhood. So what's the example I show here? All right, well, let's take the people with hypertension, and let's type them for a marker that's up there, and let's say that this particular variant comes in 2 flavors. It's either a green or it's purple, just for simplicity. Well, you can see the people with hypertension, the distribution of green and purple, people without hypertension, green and purple. One might argue that there's really no correlation between whether you have hypertension and whether you have the green variant or the purple variant. But let's keep going. Let's go and do some other of these haplotypes. Let's go over here. We'll do that one. Now, all of a sudden, the variant we're testing is orange and blue, and you can immediately see, wow, the people that got the orange variant are the people that get hypertension more. The people that get the blue variant tend not to have hypertension. So it doesn't mean that orange variant causes hypertension, but it does mean that there's probably a variant somewhere within that neighborhood, somewhere within that block, that might be conferring risk for getting hypertension. This is what is known as association, associating a variant with a disease. If you survey across the whole genome and all the blocks, you're doing it genome-wide, and that is why it is called a genome-wide association study. So the question was, this sounded great, and you can make really cute PowerPoint slides that show this strategy. The question was, would it work to the point that you could truly rule out certain regions of the genome and truly rule in other parts of the genome and have it make any sense at the end of the day? And the answer was, yes, this actually would work, and you could survey across the entire human genome, as we're doing here, and find places where there is high statistical correlation between having a particular variant and having that particular disease. And we were excited to see by 2005 the first successful example of this. It was actually when some of the earliest data from the HapMap project came out, came a story where age-related macular degeneration, a region of chromosome 1 was found associated, and they even figured out what the gene was. And we started to catalog this success. People always wondered, would it work? And this paper demonstrated that it did work. So we actually started cataloging it. Shown here is all the human chromosomes, and what we did for that first paper was to show by a little lollipop where in the genome was it associated with that disease. And then we just started following the scientific literature. And by 2006, there were a couple other successful genome-wide association studies, and you can see other lollipops were stuck on the genome. Well, by 2007, this just took off like mad. And all of a sudden, every single time we looked up in 2007 and in 2008, more and more and more and more papers were being published that described successful genome-wide association studies. These were for diseases like hypertension, diabetes, cardiovascular disease, all the things we care about. And all of a sudden, we were going from having not knowing anywhere to look in the genome to now having very discrete regions of the genome labeled by these lollipops that clearly indicated where one might search in greater detail to find specific variants conferring risk for disease. And that trend just continued and continued and continued. And here's the latest graphic. In 2005, the first successful study. Fast forward to now 2013, over 1,400 successful studies that have demonstrated where there are regions of the genome we want to be looking in greater detail to find the variants that are conferring risk for specific human diseases. That has been just truly remarkable. Now, the fact of the matter is it doesn't mean we have the answers yet. In fact, all we've done is we now have it down to a neighborhood that we need to now search door to door to figure out which is the variant that's the one conferring risk. But that has greatly changed the face of many studies in human genetics and has been really exhilarating to watch the potential this might have for the next phase of trying to figure out the molecular basis of human disease, especially complex diseases. Now, that was the glass half full. Let me always bring you back to the glass half empty. Guess what? We've learned something else in the past 3 or 4 or 5 years. We are now learning that increasingly, when it comes to rare genetic diseases, most of those changes, those mutations, those typographical differences are in the protein coding portions of the genome, the part of the genome we understand. We understand how to break the genome and have it not make a protein. The problem is over here, this great medical challenge for the future. Most of those variants are sitting outside of protein coding regions. It turns out that those are probably changes that are affecting our circuitry. And we don't know how to interpret that as well yet because we don't know how all of that works. And so this is going to be a huge technical challenge and a knowledge challenge, but it's one that we are very motivated to figure out because we need to figure out what variants are conferring risk for these complex diseases. And then we want to understand what we might be able to do with that knowledge. And so it's going to be a hard road ahead, but at least we're in a place of being in a much better position to try to study it. In order to study it, what we clearly are going to have to do with our 1,000 people with hypertension, our 1,000 people with Alzheimer's disease, our 1,000 people with diabetes, is that we need complete inventories of all of their variants because we're going to really need to tease out which of the ones are truly conferring the risk. And in order to do that, we're going to not just be able to do genome-wide association studies the way we've done them in the past. We're really going to have to sequence lots and lots of people's genomes completely to be able to get complete inventories of all those variants, which leads me to this third step along the way, and that's routinely being able to sequence whole genomes. Now here, I once again have some pretty exciting things to tell you because it has been truly remarkable what has happened in this field. So far, what I'm going to tell you about has mostly affected these first 3 domains, but the fact of the matter is, with time, as you'll see, it'll be affecting these other domains as well. Well, what's the origin of these advances I'm about to tell you about? Well, remember I told you on the day the Genome Project ended 9 3/4 years ago, we published this strategic vision for what was needed beyond the Human Genome Project. And we wrote a lot in this article. But one of the things that we wrote, which was in many ways audacious and in many ways remarkable that we were willing to put this into print, is that we called for technological leaps that seem so far off as to be almost fictional, but which, if they could be achieved, would revolutionize biomedical research and clinical practice. And the example that we gave in 2003 was the ability to sequence DNA at costs that were lowered by 4 to 5 orders of magnitude than the current cost, allowing the human genome to be sequenced for $1,000 or less. Now, why was that such an audacious thing to put with your name on it, my name was on that, into the scientific literature? Well, the reason it was so audacious is the day this came out, we had just finished sequencing our first human genome at a cost of $1 billion, roughly. And here we were proposing to take on the challenge of lopping 6 zeros off of that figure by incrementally improving technologies for sequencing DNA and delivering on something that was quickly referred to as the $1,000 genome. Well, the $1,000 genome became a battle cry for the scientific community. Our institute gave out grants to all sorts of people to come up with crazy and wild ideas for sequencing DNA. And the notion was we wanted to replace the factories that sequenced the first human genome as part of the Human Genome Project and replace it with some fancy schmancy new technology, a nano this, a micro whatever, something that would reduce the cost substantially and allow us to generate a sequence of the human genome for $1,000. Now, I will tell you, I've been involved in genomics for 25 years. I could tell you about all sorts of remarkable technological advances, but no doubt, the number one technological advance that's happened in genomics in 25 years has been not the development of 1 or 2 or 3 or 4 or 5, not 6, 7, 8, but 9 or more new technologies. These are just available instruments today that allow you to sequence DNA at remarkably reduced costs and with remarkable speed. It has been truly revolutionary what has transpired, and these technologies are completely changing the face not only of genomics, but are changing the face of all biomedical research. Now, have these new technologies reduced the cost of sequencing? Absolutely. It has been truly mind-boggling. Let me show you some real data. If you ask me the question, what was the cost for sequencing the human genome over different periods of time? If you went to sequence the human genome, we actually collect that data because we support groups that do nothing but sequence human genomes. Let me remind you of a concept, Moore's Law. Probably many of you have heard of Moore's Law. Moore's Law is the law of the computer industry that basically says that compute power doubles about every 18 months. And if you talk to experts in technology development, they say nobody keeps up with Moore's Law except for the computer industry. They have set the standard and nobody beats them. Let me show you some data. So shown here is a graph that depicts the cost for sequencing a human genome starting at about 2001 in green. And notice that the y-axis is logarithmic, and notice in white is Moore's Law. So if you follow Moore's Law, you rock, okay? Well, our sequencing centers for a long time kept up with Moore's Law. They rocked. And then right here, they switched to these new technologies that have been developed for sequencing DNA, and ever since then, they have blown Moore's Law out of the water. Truly, truly remarkable. And in fact, if you ask me today, what does it cost to sequence a human genome? I will give you a very accurate answer. As of today, it costs exactly that amount to sequence a human genome. It's not quite at $1,000, but we're going to get there really soon. A couple $1,000, maybe a few thousand, depends exactly how you calculate it. But it's not just the cost of sequencing. It's actually the speed at which you can sequence a human genome. To put it in context, when we sequenced that first human genome as part of the Human Genome Project, we were actively sequencing for 6 to 8 years, and it cost $1 billion. The day the Human Genome Project ended, we did some calculations. If we would have said all these sequencing groups go back and sequence the second human genome, they came back and said if you gave us the money, it would take us about 3 to 4 months. It would cost about $10 to $50 million. Pretty good improvement, but still slow. Today, using various technologies, I hold in my hand one of the fancy little chips that can be used for sequencing a human genome with one of those instruments. And with one of those instruments and a chip like this, you can now sequence a human genome. It'll take you about 2 to 3 days. I actually know for a fact that that'll be reduced probably to a day within about the next 6 months, and it would cost something on the order of $4,000, $5,000, $6,000, in some places maybe even less than that. Truly, truly remarkable, completely game-changing. And you know what? It's only going to get better. We already know there's new technologies coming on board. For example, there's a fancy new thing called a nanopore, which is basically a molecule that sits in a lipid bilayer, and the DNA strand gets pulled through it, and letter by letter gets read off as it gets pulled through. It's just completely way cool, all right? And it even gets better than that. Allegedly, and I don't know if this is true, but allegedly a company is about to commercialize instruments for using this technology, and one form of the instrument will be a little USB device that plugs into the laptop, and in a day will read out a human genome sequence, if it's true. Which is truly remarkable. And do you know what else is incredibly remarkable? Is I asked the company, and this USB device will work equally well in a Macintosh or PC computer. So they've thought of everything. So stay tuned. I do not worry anymore about getting to the $1,000 genome. It is not what keeps me up at night. But you might ask me, what keeps me up at night? And I will tell you, this is what keeps me up at night. Routinely being able to analyze this sequence. We have gone from having a situation where we are data poor, analysis rich, because before we had the first genome sequence, to now having a circumstance where we can generate so much data, but we can't analyze it fast enough. And this is affecting every single area across all these domains. You know, the current bottleneck in all of biology, but certainly in genomics, is caused by our own successes. We are victims of our own success. We develop technologies for sequencing genomes, and we haven't kept pace with our ability to analyze the data. This is what it's like. It's like trying to assimilate as these data get spewing out of these machines. We can't capture it all. We're overwhelmed by this. We are big data people. You know, in the past, it was the particle physicists, it was the climatologists, it was the astronomers. They were the big data people. Biologists, we had little data. Now, we've got big data. And this big data is now a bottleneck. We don't have enough servers and processors for handling all the data. We don't have enough computer tools that allow us to analyze the data robustly. And for any students in the audience, pay attention. The workforce, the next generation, we need many, many new people trained in new ways to be able to be biologists and clinicians and physician scientists and so forth. And so we're tackling this at NIH and other places. But it's not the only bottleneck. It's not just all a compute issue. There's also an informational problem here. I want to be very candid and clear. We have fancy methods for sequencing a genome. I could sequence any one of your genomes in 2 to 3 days. And I could even get past the computational bottleneck, and I could even read out the list of your 3 to 5 million variants. But the truth of the matter is, if you were a patient in a hospital, or you were a subject in a clinical research study, and I had those 3 to 5 million variants, when I rounded on you in the morning, you know, I would be puzzled by most of those variants. I don't know what most of them mean yet. We don't have information. And most of the time, we're going to feel like this, like, wow, there's the list, but I'm really not sure. So we got a challenge ahead of us, but we're going to deal with it. It is interesting. Harold Varmus, Nobel Prize winner, previous director of the NIH, now head of the Cancer Institute. He wrote an editorial piece a few years ago where he talked about how physicians are still a long way from submitting their patients' full genomes for sequencing, not because the price is high, but because the data are difficult to interpret. That's really where we are now. A colleague of mine wrote a piece also recently talking about the $1,000 genome, but the $100,000 analysis, and you sort of laugh, and then you say, yeah, but it's actually not very funny. We got to solve this too, and we will. So those are the 5 steps I wanted to tell you about. I recognize those aren't the only 5 steps. There are other things you're thinking about, especially the more clinically oriented domains, whether it's new diagnostics, or it's new therapeutics, or it's new preventative measures. All these are going to happen, and there's probably things that none of us have thought about at all. And that are anticipated and that are profoundly exciting. And that now leads me to sort of the last segment of my talk. I promised you I would take you to the future, and I want to do that now. And I would tell you that the future is very much going to be driven by technology. And maybe that's obvious to all of you, but I do want to remind you that technology just drives science. You know, whether it's, when we came up with the telescope, that's what drove our understanding of astronomy. We came up with the microscope, that's what drove our understanding of cell biology. We came up with the imaging technologies like CAT scans and PET scans, that's what drove radiology. And these new sequencing instruments for sequencing genomes are driving genomics at an incredible, remarkable pace. And they will for the foreseeable future. Now, one of the immediate things I want to do is to tell you about the next 20 years at a very high level, then I'm going to give you some specific examples. At a very high level, let's fill in the rest of this chart I showed you earlier. How are things going to advance over the next 20 years? Well, what's the rest of this decade going to look like to 2020? I want to be very clear. We're not going to change the face of medicine by the end of this decade. The center of gravity is going to move rightward. I think the next decade is going to be very much about understanding the biology of disease, as well as the remarkable continued advances in understanding how the genome works. With that will come some clinical advances, but real clinical advances are hard, and they take a long time to fully operationalize. They're coming beyond 2020. I just want to manage expectations by reminding you that I think they'll be there, but I don't think they'll necessarily be there just before the end of the decade. But there will be considerably more achievements in these clinical domains before the end of the decade. How is that going to be accomplished? Well, we're going to sequence a lot of people. No, not just the people on this slide. No, not even the people on this slide. In fact, not even just the people on this slide. We now think already, we believe before the end of the next few years, we'll have over 100,000 people's genomes will be sequenced. Most of these are taking place as part of major clinical research studies, but increasingly these will be done as we figure out how to operationalize this for practicing clinicians. And there's many studies that we're doing to start to look at that. Critical to this is as these genome sequences get generated, they need to be shared widely to the whole research community, because the greatest power of genomics and our understanding, especially of the genetic basis of common diseases, is going to be by having lots of data available and having a lot of scientists looking at that data. And so increasingly, sequence will be streaming into public databases for scientists to be able to access and to be able to utilize in a fashion to allow them to enhance their studies. What are some specific areas that in particular a funding agency like ours is focused on? Well, let me remind you what some of the key ones are. Using these powerful new sequencing technologies, we need to fill in this pie chart, and we're going to do that. We have a program that we've just launched. The groups are going to focus on accelerating the rate at which we can identify the genetic basis of these rare diseases and fill in these remaining sectors of this pie chart. We similarly have programs where we're going to go from these lollipop regions of the genome to drilling down and really figuring out which variants are the ones conferring risk for Alzheimer's disease, for diabetes, for mental illness, for asthma, for cardiovascular disease, and so forth. But those are all big, long-term projects, and I'm not claiming that they're not anything other than a considerable amount of additional work. A logical and fair question some of you might ask me is, what's some of the lowest hanging fruit out there? What are the things that might actually change medicine before the end of the decade? What are some of the greatest things you think are going to happen the soonest? And if you ask me that, I will give you one immediate answer because I firmly believe it, and that comes in the area of cancer. And the reason I pick cancer is, let me remind you, cancer is a disease of the genome. The reason that cancer forms is because the genetic blueprint has broken in some way, making cells grow out of control. And guess what? We now have the ability to open up a cancer genome and see where the misspellings are, where the changes have taken place. And through efforts such as the Cancer Genome Atlas, which is NIH's big effort in sequencing tumors, sequencing cancer specimens, but this is going on around the world. At a ferocious pace, we are now at a point in time where many, many different kinds of cancers are having their genome sequenced, and it is revealing remarkable things about and changing our understanding about cancer, and in some cases leading to new approaches in cancer. So today, cancer diagnostics mostly takes place by having pathologists look under a microscope and say, okay, this is this kind of cancer. But many times we're wrong of knowing whether by this view of this cancer, whether to give a patient this therapy or that therapy or some other therapy. And in the future, and by the future I mean it's already happening for some cancers and it absolutely will be happening for others in the next 5 years, we'll continue to have that pathologist look at that specimen and try to figure out exactly what kind of cancer it is. But more importantly, we will also use a sequencing instrument to make a genomic map of that cancer, and from that we will get information about the best course of treatment, and in some cases we're learning about new avenues for developing new treatments. This is going to be game-changing in cancer, and I think it'll be one of the first areas of true genomic medicine being implemented. What's a second example of low-hanging fruit, things we're already seeing? It's in the area of therapeutics, medicines. I'm not sure most physicians think a lot about genes, but they're going to start to now. And one of the ways they're going to start to think more and more and more about the genome is because they are recognizing that every patient is different, that every patient has its own unique genomic information, and the fact of the matter is everyone responds differently. And everyone responds differently to not only things in life, they respond differently to medications. Because the fact of the matter is every single medication that comes to market that you can get in your pharmacy, every one of them work. They just don't work in everyone. And the fact of the matter is, a major reason why they don't work in everyone is because each of us metabolize drugs a little bit differently. And guess what? How we metabolize drugs is partially an effect of genetic variants that we have in our genomes. And so the idea of pharmacogenomics, pharmaceuticals and genomics coming together, is going to create a new era for many kinds of medications. It's already true for a handful, but that list is going to grow with time, where we can take individuals all with the same diagnosis, all the same disease, but we can, in advance, stratify them by testing their DNA, testing their genome, and figure out those people that won't respond to this medicine and those that will respond poorly to the medicine, and don't give them the medicine. Give them a different medicine. And only give medicine to these individuals who are going to respond without having side effects. And this isn't science fiction, this is real. It's real for a handful of medicines now, and increasingly it's going to grow in list. And all of us have the experience of getting a medication that makes us sicker or has no effect, and a lot of that response is due to medication. So these are the kinds of new initiatives that we are now putting into play. I just told you about pharmacogenomics. We have other pilot efforts that we in particular are setting out research projects on. Some of these are actually taking clinical care and using genome sequencing as part of that care, but doing this in a research setting to figure out how we're actually going to carry that out. We have various demonstration projects that we're now embarking on to try to test some of these things out and help different groups learn how to do this from others. Another area that we're certainly now doing research in is in the area of newborn screening. Every human born in America, and actually most developed countries, within a day of birth, they get a little heel stick, and they get a little blood taken from them, and they get a battery of about 25 to 50 genetic tests done. But what might that future look like when we now know the genetic basis of 4, 5, 6, 7, 8,000 rare genetic diseases? And maybe it'll just be cheaper to sequence their genomes than to test them for 25 or 50. But there's lots of issues there. Who should have that information? What do we do with that information? How do we actually operationalize that? So we have research studies that we're now starting to investigate. What might a future look like with newborn sequencing? Do we want to go there? What might we learn? How would we actually use the information? And similarly, there's a lot of information that we need to learn about all these variants and which ones are relevant clinically. I showed you the cartoon earlier of the person sitting there at the bedside of the patient not knowing what any of this means. And I will tell you as I've traveled around that truly we need a better clinical system for assimilating all this information, especially with the onset of better and better electronic health records. We simply need to provide practicing healthcare professionals much more information about genetic variants so that in their brief encounter with a patient, they can quickly figure out which genetic variant might be relevant, which one might not be. And we need to have them that be curated so they can look that up and immediately see what to do with that information. So let me just tell you one other really cool example before I truly do wind down, because some people think this is also a point I probably need to make. I don't mean to imply for a minute that the only relevant thing in human disease is genetics. I fully appreciate that there is a role for both the genome and also the environment in understanding human disease. And I showed it in this graphic where I clearly showed the environment. The fact of the matter is the main reason I've emphasized genomics is because, one, it's what I do, but two, because there's been this remarkable advance in genome analysis technologies. And the fact is that's lagged behind considerably technologies for monitoring the environment, although those are improving. And I think over the course of the next decade, we'll get better and better ways of monitoring what we eat and what we're exposed to and so forth. But let me just quickly tell you a story that I think is so cool, because it's using genomic technologies to actually also monitor the environment. And in fact, the environment is the most intimate environmental exposure that we have because it's in us and on us. It's something called the microbiome. You know, when we're born sterile. We have no microbes living in us and on us. We are born essentially sterile. But within a matter of weeks, we are quickly colonized by many, many, many microbes, bacteria, fungi, and viruses, and so forth. And in fact, each and every one of us has about 100 trillion microbes living in us and on us. We remember we're only 10 trillion. We are outnumbered at the cellular level 10-to-1 to microbes. You are a minority owner of your little ecosystem, okay? You probably never realized it, and you probably all want to go wash your hands right now. But get used to it. Collectively, that community of microbes that outnumbers us, our most intimate environmental exposure is called the microbiome. One other statistic, and then I'll tell you the way cool part. 90% of those microbes that are living in you and outnumbering you have never been isolated, never been studied. We don't even know what their name is. We're blind to them because we don't have a laboratory method to isolate them. Only about 10% of all those microbes do we even know exist. And when you get cultures done, they only grow at about 10%. But now, with these new sequencing methods, we can actually go through and we can actually study that other genome. Because now, instead of actually isolating those organisms, we can just isolate material from you, purify the DNA, sequence it, and then inventory what we see at their DNA level without actually having grown those organisms. This has led to a big project called the Human Microbiome Project, which is just winding down, but it is a project to catalog these microbes that live in us and on us at different sites, in your mouth, in your ear, in your nose, on your skin, and so forth, and then be in a position to figure out how those community of microorganisms change in health and disease. Because it's not all, most of the time we get along great with our microbes, but sometimes that little equilibrium gets off and the microbiome could actually be detrimental. And we want to be able to understand that and study it. It is absolutely going to be a major part of biomedical research in the future. And it's an area that is going to change many aspects of clinical microbiology. Once again, I'll take you to that pathologist who right now might look at Petri plates and might look under a microscope at the microorganisms, but he's only seeing 10% of what's there. But in the future, he'll still culture those 10% and study those 10%, but he'll also sequence what's there, and with that, he'll get much better views of the full inventory of microbes and how those communities are shifting with and without disease and what role they might play in disease. So it's just sort of a way cool way of thinking of it, that we're using genomics to also monitor our environment. And it's a growth area in biomedical research I want to tell you about. Okay, let's take a step back. I've gone through a lot of material, I know. The truth of the matter is when the Genome Project started, we had a vague idea that someday, this might be relevant for medicine. I think by the time the Genome Project ended, we started to get some feel for that there was good use of genomics for health and healthcare, but it was still pretty fuzzy. I would say that when we published our strategic plan in 2011, it started to come into focus. And while it's not totally in focus quite yet, I really do believe by the end of this decade, many aspects of it are going to be crystal clear. And we won't have all the answers, but we'll have a lot of the knowledge of what we really need to do. With that said, there's lots of other things I'm sure we could talk about. There's all sorts of issues at the intersection between the genomic advances I've described and the ethical, legal, and social implications of what we do. There's many societal issues, and I wouldn't be surprised if some of the questions you ask me will, as often is the case in audiences like this, will relate to that. And there really are many great questions. We actually study a lot of these, and we give grants to study a lot of these ethical, legal, social implications. And I will just tell you quite candidly, while I'm extremely enthusiastic and I'm extremely excited about the future, I don't pretend to know all the answers, and I don't think the genomics community knows all the answers. I can't help but quote Albert Einstein. This is actually a sign I keep immediately above my door in my office. And it really is true about genomics. I mean, truly, if we knew what we were doing, we wouldn't call it research. And we still everything I'm describing to you is still research. It's exciting, but nonetheless, so much of operationalizing this is going to require a research agenda. So let me just leave you with a quote before I just tell you about two more things. And that quote really reflects my view of the field of genomics. It's a Winston Churchill quote. Winston Churchill said that a pessimist sees the difficulty in every opportunity. Genomicists, we are not pessimists. You know, we see opportunities, and we did the Human Genome Project. We see opportunities. We're getting close to a $1,000 genome. We are optimists, and we see an opportunity in every one of those difficulties, every difficulty we encounter. And I think that very much flavors the kind of field that I get to represent. So just in the last 2 minutes, I'm just going to tell you two other things. You might say, wow, you are very enthusiastic. What fuels your excitement about genomics? I could come up with lots of examples, but I just can't help but tell you how proud I was that Time Magazine at the end of 2012 put out a whole set of top 10 lists, top 10 story in politics, top 10 stories in business, blah, blah. They put out their top 10 medical breakthroughs of 2012, the whole world of biomedicine, top 10. We got 5 of them. They talked about cancer genomics in a project that one of our big funded centers is involved with. Just exactly what I told you about, sequencing, in this case, pediatric tumors. Number 7 or 6 on their list was a story about breast cancer genomics, the exact project I told you about, the Cancer Genome Atlas. Number 7, speeding DNA-based diagnostics for newborns related to newborn sequencing using these two technologies. Remarkably incredible. That, again, high on the list. Human Microbiome Project I told you about, number 2. Number 1 medical breakthrough for 2012, according to, was the ENCODE project, this interpretation developing a catalog of how the human genome actually works. 5 of the top 10 were genomics. And if you think that doesn't make someone like me or other members of the genomics community proud, you're missing something. Lastly, let me just tell you about, that was last year. What about this year? I want to extend an invitation to all of you. This is going to be an incredibly exciting year for genomics, celebratory year. 60th anniversary of the Watson-Crick discovery of DNA, pretty remarkable. 10th anniversary of the completion of the Human Genome Project, April of this year. We are doing lots of things to commemorate the 10th anniversary of the Human Genome Project. If you're interested in following some of those things, go to this URL, genome.gov/HGP10. There's going to be a symposium and lecture series, all of which we will put up on the web. We actually have a YouTube channel called Genome TV, and we post lots and lots of videos, and all of our major events, which are described here, will be posted. So any of you who thought what I talked about was cool, you can get even way more cooler stuff if you listen in on some of the things that will be happening throughout this year. And here's my invitation. My invitation is I hope all of you will come down to Washington, D.C., because we have a partnership with the Smithsonian's Natural History Museum, where we are jointly putting out a new exhibition on human genomics. That exhibition will open in June of 2013. It will be in Hall 23 of the Smithsonian Natural History Museum, right next to the Hope Diamond. You can't miss it. Just go to the Hope Diamond and take a left, okay? This is the exhibition, Genome Unlocking Life's Code. I just reviewed the 95% design. It's been one of the most fun things I've done in my life of co-designing an exhibition. It will be at the Smithsonian for 12 months, starting in June, after which it will tour North America for 4 to 6 years. And it's something we've done in partnership with them, and we think this is just wonderfully exciting. And with that, I will end. I thank you for your attention. I'm happy to answer any questions.
Hold up your questions, please, and let's save your applause. We have about 10 minutes for questions and answers, so please hold your questions up so they can be collected.
We can turn the lights up if we're going to ask questions.
Right, I'll work on that.
Sure.
Okay, lights, okay. They will come up. Okay, Dr. Green, what are your thoughts about privacy issues and other dangers that may result from wide sharing of an individual person's complete genome sequence?
So let me repeat the question, which, as anticipated, is both a good question and a question related to some of the ethical issues, and it related to privacy. There is obviously quite a lot of intimate information buried within our genome sequence, especially as we learn more and more about genome variants each of us might have and what it might mean. And what do I think about these things in terms of risk to privacy? And what I would immediately say is, this is something we are actively studying, both with respect to what the privacy issues are, what people's tolerance of this might be, ways to protect individuals, and so forth. Also related to this is people who are involved in research studies, making sure they have been appropriately consented, they have a full understanding what the implications might be. For many of our studies, where we are getting information about individuals' genomes and information about their clinical characteristics. Those sorts of data are put in databases that are protected from the point of view that only scientists who have applied for access to them can get it and have to abide by confidentiality issues. But this is going to be an ongoing, and many of the issues I talked about around newborn screening and newborn sequencing immediately comes to the fore about, well, who has access to that information? If all newborns have genome sequence available, how do we protect individuals? This is sort of this classic two edges to the sword. On the one hand, powerful opportunities. On the other hand, we don't want to have this used in a detrimental way. It's an ongoing, we have to constantly be looking at this and constantly be aware and always give people the protections that they request. And we certainly find, as we study this, a wide range of people's concerns about this. And I will tell you that in today's world, especially the younger generation who are heavy Facebook users, who I'm amazed how much personal information they share on Facebook, they tend to be much less concerned about some of these issues about privacy, about their genetic information. And there's people obviously at the other end of the spectrum who are very concerned, and we have to honor that as well. So it's really trying to define this. We are very aware of this, and that also includes investing money to research this, to understand it. We can't do this in an insensitive way, and we won't. Well, you got a lot of questions there.
Oh, yeah.
You got to sort them all out. Go ahead.
We won't get through them. I'm trying to get through as many as I can. Are you concerned that recombinant DNA technology could result in serious problems such as the development of a superbug or other organism that could drastically alter the ecological balance of the Earth?
Wow, that's a heavy one. By the way, there's probably very little that I have spoken about that hasn't been, that question mostly relates to recombinant DNA concerns that have been in place ever since the molecular biology revolution. of the 1980s. And probably as much of what that's being described could be genetic and genomic manipulations of microorganisms or selected animals as part of agriculture, or sort of livestock, or you think about genetically modified fish and so forth. I'm not personally as concerned about some of these things as others might be. These have been studied and monitored extensively for many years by the NIH and continue to have many steps of oversight in these things. There was certainly this year an interesting flurry of concern around some studies that were being done, H1N1, flu, and concerns about some implications of that. And at the end of the day, yet, again, oversight is being put into place to properly ensure that we don't have detrimental consequences. I think it's something for us to be aware of, and we can't ignore this, and NIH certainly is not. In the grand scheme of things, I think some of these concerns are far greater than what the scientific basis of them would indicate.
If a person has been diagnosed with a rare genetic disease, specifically a deletion of PMP-22, which results in a rare form of muscular dystrophy, where does one go for medical advice and consulting? Most physicians are uneducated in this area and can offer little help.
I'm not familiar with the particular gene you're talking about there, but this is a very common question about rare genetic diseases that you're absolutely right. These extremely rare diseases, a typical practice physician may not know. There are various resources that are available, including some that we have at NIH. If you sent me, whoever asked the question, an email, I could easily refer you to staff that will point you to all sorts of places that you could look at. We have an NIH sign called the Office of Rare Disease Research, which deals with exactly these issues, both for referrals and patient groups and connecting people and scientists and physicians who might have expertise in those diseases. The real answer to the question is almost for certain. There's somebody out there. If the gene has the name, there's somebody out there who's an expert, and it's just a matter of connecting with individuals. That said, it doesn't always mean just because we know the genetic basis there's a cure or there's a treatment. In some cases, sadly, we may not have that yet, but there are ways to network to experts.
What safeguards do you recommend to protect individual rights and individuals' privacy rights?
So the first thing I would say is that nobody is forcing anybody to participate in genomics research, and nobody is forcing you as part of your clinical care to have your genome interrogated or sequenced. So first thing is you can decide what you want to do. And so I think that's the first and foremost the way to do it. And there are laws that exist in some cases. There's something called the Genetic Information Non-Discrimination Act that Congress passed a number of years ago that at least protects you in certain areas that you can't be discriminated on. We're certainly working to try to even have stronger. And there really are actually very few examples where this has been in fact, some people believe there's almost no examples where this has really materialized in a serious way. There is concern, and we actually continue to study this to figure out whether the laws are adequate, to see what other safeguards might be put into place, and so forth.
A couple of related questions.
Sure.
Whose genome was originally sequenced? And related to that is, since everyone's genome is different, how did the Human Genome Project choose whose genome to sequence, and how accurate is it as a reference sequence for the diversity we see in human populations? And also, are there different reference sequence for different populations for regions of the genome that do vary significantly between populations?
So I think some scientist wrote that one. That was a great question. So there are lots of sub-elements of that. Let me try to dissect them if I'll try to remember them all. But they allude to one very fundamental truth, is that we sequenced the first human genome. I sort of lied. It really wasn't a human genome that we sequenced. What we sequenced was a reference of 3 billion letters. Each of us is really 6 billion letters, so the first thing is we didn't really sequence a human's genome, we sequenced a human genome, if you see the difference. The other thing, the answer is we didn't just sequence one person. It ended up that for a variety of just purely logistical things as much as anything else, we sequenced stretches that were from one person, then the next stretch was from another person, and maybe that next stretch was from that first person, and the next stretch was... It was sort of a quilt, a patchwork, if you will. And so at the end of the day, when you say, here is the sequence of the human genome produced by the Human Genome Project, it is just a hypothetical person patched together of little bits and pieces from many, many different people. But it's 3 billion letters as a reference, which is roughly 99.9% identical to everybody else. So for the purposes of having a reference, it was good enough. One of the sub-questions in there was, well, is there more than one reference sequence? In fact, that's one of the things being discussed now, is that what we really need is not just a hypothetical. We need a set of really, really good reference sequences. Some of these are being generated. And as the question also alluded to, probably for different individuals from different geographical origins, they're going to vary a little bit more, a little bit less, depending upon exactly what comparisons you do, and we probably need to have that representation in the set of references, which is exactly why some of these projects like 1,000 Genomes and HapMap project I told you about sampled across the world to try to capture that diversity and represent that appropriately. So all this is, so it is an oversimplification to think that the Human Genome Project produced a sequence of a person. It didn't. It was just a hypothetical reference of 3 billion letters. That's 99+% the same as all humans.
I'm trying to bundle questions so we get as many answered
Sure.
as we can in the next, looks like 2 minutes. Within the NIH and your specific domain, what is your plan for maintaining continued funding given the current physical climate? And related to that is, how does the NIH partner with universities, for-profit businesses, and other organizations to achieve your research institute's goals for knowledge and use of the human genome?
So both great questions. What do I do to try to ensure funding? First, I vote. Second of all, I tell all of you to vote. Actually, in all seriousness, if you think this is way cool, why don't you go write your congressperson and your senators and tell them how incredibly important you think this is for the future of human health and well-being. Because at the end of the day, we're completely dependent upon Congress in particular and the White House making decisions about our levels of funding. It is a very precarious time. It is I hope I have convinced you that we are at a very bizarre point in our history where I hope I convinced you that some of what I'm describing scientifically is the most exciting things going on ever in terms of opportunities to improve human health at the very same time that I spend the majority of my time worrying about our funding. You would think we should be pushing the accelerator incredibly hard and funding this work at an incredibly fast pace, but in fact, I'm worried about a fiscal cliff and various other things. So it is a strange time to be living in, but we're a democracy here. We need to influence this. So I hope all of you could become excited about this and try to influence the people that make these decisions. The other part of the question is, how do we interact with universities? How do we interact with companies? We give grants. There's, sitting right here, some major grantees of our institute, some very prominent genomic scientists that work at Penn State. They get money for us to do their work. There's some of the more scientific leaders of this field. Similarly, companies, for-profit companies, we give grants to as well. We would not be close to a $1,000 genome right now if we didn't develop partnerships and in some case give grants to for-profit companies to help operationalize these technologies so we could sequence DNA better and better. So this is all very collaborative, both with academia and with for-profit companies as well.
You all never let us down in the really great questions that you write. We didn't have time to answer all these questions, so I will copy them and I will give them to our speaker, who, let's give a big warm welcome and warm thank you to Dr. Eric Green.